Hello, everyone. My name is Alicia Milliken with NAMA. It is my pleasure to introduce tonight's speaker, Dr. Amy Honan. Amy is a mycologist studying fungal biodiversity, evolution, ecology, and biogeography. For her BS, she studied the population genetics of the fungal root pathogen Armillaria. Her master's thesis investigated the evolution of the sapotrophic fungal genus Tetrapyrgos. Amy's PhD dissertation explored the ecology, evolution, and diversity of the stalked puffball genus Tulostoma. She's the co-founder of the Crested Butt Botanic Gardens and teaches mycology and fungal ecology for Oregon State University. Her current work involves documenting fungal biodiversity and ecology and understanding heavy metal tolerance in Solus. Tonight, Dr. Honan is presenting for us Stalked Puffballs and Other Desert Fungi, exploring the fungal diversity of desert ecosystems. Thank you so much for being here with us, Amy. I will turn the floor over to you.
Thanks, Alicia. And I wanted to thank NAMA and Trent for putting together these webinar series. I've been having a lot of fun watching them, and hopefully you guys are enjoying it as well. So Alicia already went over this, but I'll just tell you a little bit about myself. I started out at my bachelor's degree at Western Colorado University in Gunnison, Colorado, looking at Armillaria. So I was in charge of culturing these guys, and that was kind of my introduction into fungi. After that, I moved to San Francisco State, where I studied the mushrooms of Southeast Asia under Dennis Desjardins. So we got to travel all over Southeast Asia, meeting all kinds of tremendous mycologists. And I studied the genus Tetrapyrgos. And so if you were lucky enough to see Brian Perry's talk the other day about the beauty of mushrooms underneath the microscope, this is one of those small little saprotrophic fungi that I think is beautiful to look at, but under the scope, it's really spectacular. It has all these different cystidial characteristics. So they're really fun to look at, and they have really cool spores. After my master's, I went to the University of Washington and I studied the genus Tulostoma. So we'll be talking a little bit about Tulostoma this evening. And then currently I teach for Oregon State in their online program, their eCampus program, which enables me a lot of flexibility so I can travel a lot. And I'm actually based in Colorado. So I'm based in a little ski town called Crested Butte, and I've established a botanic garden there. So let's talk about why Tulostoma. There's so many different cool fungi. Why would I focus on this weird, not really maybe that charismatic fungus? And so the story kind of starts where I live in Crested Butte, which is this little dot here in Colorado. So my house is actually at 10,000 feet. And this is what I look at when I look out my window. So that mountain right there is Mount Crested Butte. And so I'm immersed in this mountainous, mostly alpine kind of habitat with these mixed coniferous forests and these alpine meadows. And I, for a long time, taught down in Gunnison at Western Colorado University. And one spring, I was teaching a spring fungi class. So in my mind, I was thinking about snowbank fungi. And the snowbank fungi hadn't started to pop, so we were collecting more close to campus down in Gunnison. And the very, very first mushroom I found on the very first day of this class was Tulostoma. And Tulostoma to me is more of a desert fungus. So it really started making me think about the ecology of my area. Because Gunnison is only 30 miles away from Crested Butte, but it's a much different ecology. So if we're thinking about the Up Valley, the Crested Butte area, we're in those subalpine forests, those mixed coniferous forests and alpine areas. But if we go down valley, we're in these more sagebrush parks and grasslands and sort of a steppe habitat that looks a lot more like this, where you can see these grasslands and sagebrush habitats as we look towards the mountain. So that was pretty cool for me to recognize, oh yeah, I do have different kinds of ecosystems right out my back door that I wasn't really contemplating before. And the other stuck puffball that I was familiar with was Bataria. So if you're familiar with Bataria, depends on which authors you go by, but there's about 3 species of Bataria. So I naively thought it would not be hard to identify Tulostoma. When you look at species Fungorum right now, there's about 289 species. which blew my mind. I didn't realize there was that kind of diversity, especially in sort of an arid adapted species. So then I started looking at the morphological diversity of this group, and what really bowled me over was the spore diversity. So if you look at these are all different spore morphologies within this genus Tulostoma. So the spores go from completely smooth to slightly roughened to all kinds of different spines to ridges to reticulate nature or even spiral. And if we look at the distribution of Tulostoma in North America, what you're seeing here is that kind of the center of diversity of this genus lies within the southwestern US. And so I started realizing that this would be a pretty cool project to try and understand the diversity of this group. So first we want to think about the ecosystems of the American Southwest. So we have montane forests and we have rivers and streams, but where this mushroom occurs is more in these more arid environments. So these pinyon juniper woodlands, these grasslands and shrublands, and these deserts. And so these historically might be habitats that we're not really thinking about fungi existing in or having a ton of diversity. So we don't, as mycologists, we don't necessarily spend a lot of time looking for fungi in these types of habitats. So if we think about the ecology of North America and we think about North American deserts, they actually take up a lot of surface area within North America. They extend from BC all the way down to Baja California and north central Mexico. And deserts, as you might imagine, are distinguished by aridity. They're pretty dry. They have maybe sparse vegetation, so they have unique shrubs and grasses and even cacti, and generally lack a lot of trees compared to a forest ecosystem. They can have really cool relief patterns, like in just west of where I live, we've got the Colorado Plateau that's got mesas and spires and really cool red rock formations. And the soils of deserts are generally dry. They lack a lot of organic material. They don't receive a lot of rainfall, and they also can have some temperature extremes. So especially the southern deserts can be really hot. And so all these conditions increase evaporation rates that contribute to their aridity. So if we look at some of these habitats across North America, we've got the Great Basin Desert, which is a desert ecosystem, but also a steppe ecosystem. So it has some shrublands and grasses. We've got the Mojave Desert, where we do see some grasslands and shrubs, and we've got some sand dunes in there. We've got the Sonoran Desert with their characteristic saguaro cacti. We've got the Colorado Plateau with their characteristic red rock formations. And then we've got the Chihuahuan Desert further to the south that the Sky Islands are in. And so interspersed in these desert habitats, we also have some grasslands and different ecosystems. And if you listen to Edward Abbey, he's going to tell you that it's been said that everything in the desert stings, stabs, stinks, or sticks. But I'm here to tell you that it's also very beautiful. So here's different images of the Colorado Plateau, and this is one of my stomping grounds. Like I said, I live at 10,000 feet, as you might imagine. Historically, we've had some very, very long, cold winters. So this is a place that we like to escape to in the springtime to kind of thaw out and get some sun and kind of act like a lizard after a long winter. And if we look at deserts on a global scale, deserts cover probably or approximately 1/3 of Earth's land surface. So about 33% of Earth's terrestrial ecosystems are considered desert. And the desert is intertwined with different grasslands. So if we look at North American grasslands, we have the Great Plains, which take up the central portion of the US, and then we have other smaller grasslands interspersed, such as Mediterranean California, and then some southern arid highlands. So in this ecological diagram, you'll see that folks have sort of interspersed grasslands and desert ecosystems because they kind of go hand in hand. So if we're looking at some of these grasslands, we can consider the Great Plains, whose dominant vegetation are different species of grasses with this great expanse of rolling hills. Then we have the California grasslands across the coastline, and then again, this sagebrush steppe ecosystem. And if we consider the global scale of grasslands, they cover about 40% of Earth's land area, or approximately 52.5 million square kilometers. So we're talking about a lot of area for desert and grasslands. And if you consider that, and this is probably a location that we don't really consider as mycologists, there's a ton of fungal biodiversity in there. And they're also, according to the World Wildlife Foundation, one of the most threatened habitats on Earth and some of the least protected. So in the US, about 4% of these ecosystems have formal protection and less than 2% globally have formal protection. And so when we start to look at factors of climate change, it's very interesting to see that a lot of the plants in these ecosystems are experiencing more rapid changes in their phenology relative to other ecosystems. So they are, they're very important and somewhat underappreciated, some might say. So if you're in these sorts of ecosystems and you're seeing fungi come out of the sand, the first thing I always think of is, what are you eating? You're in this sandy, low organic material environment. What could you possibly be eating in there? And so I always flash to this scene from Raising Arizona.
When there was no meat, we ate fowl. When there was no fowl, we ate crawdad. When there was no crawdad to be found, we ate sand. You ate what? We ate sand. You ate sand? That's right.
So as Nick Page is questioning, you ate sand? What really are you eating in there? So just a little insight into me as I walk through these sand habitats and pick a fungus. You eat sand. So let's talk about some of these fungi that eat sand. So we're going to start with Tulostoma or Tulostoma. So the etymology of Tulostoma, tylos means bladder or cushion, and stoma means mouth. And so that bladder or cushion is referring to the puffball, and the stoma is referring to the opening of the puffball from which the spores emerge. And if we think about the evolution of these guys, they are in the Agaricomycotina, and they're in the Agaricales. And if we look at the phylogeny, these numbers are always changing, but we can use these as a pretty good baseline. There's about 490 accepted genera in the Agaricales and about 36,000 described species. And if we look within this phylogeny, again, these things are bouncing around, moving around, but we've got the Tulostoma.We'll, a family within the Agaricaceae sensulatu in a broad sense. So these are related to Agaricus. And if we think about the morphological diversity of Agaricales, we have all kinds of different forms, right? We have mushrooms, things that we think of as mushrooms, but we have all these other morphological forms, right? We have bird's nest fungi, and we have coral fungi that look like they should be living under the sea, and we have stinkhorns. And so if we start to think about the evolution of these different mushrooms, one evolutionary trajectory that we're seeing is an increase in the surface area of the spore-bearing surface. So on the left side of this image, we see those clavulina-like structures, these finger-like structures, and on the tips of those finger-like structures are where the spores are produced. And so over evolutionary time, we see a branching of that structure, and so we see an increase in the surface area that produces spores. Over evolutionary time, you can see sort of a planar structure is developed, and that spore-bearing surface drops down, forming lamellae. So we've even more greatly increased our surface area for spore production. And then we throw in some cross walls and the Bolitales type fungi, and we've increased surface area. So this is one of the trajectories of evolution within the Agaricales. And there's also another one where the fruiting structure or the spore-bearing structure is enclosed. So you can think of a mushroom with the gills underneath. And over evolutionary time, the mushroom cap doesn't really open. And that's that pseucosioid form that we see in the top middle diagram. So you can see the outline of a mushroom, and the cap isn't fully opened. And so the tissue layer that's inside is sort of this mottled gill structure. So you can sort of still make out gills, but it's evolving or on this way to being a completely enclosed structure, which is the gasteroid form. So we see this evolutionary trajectory happening multiple times over the evolution of fungi. And here's just another diagram of that happening. So we can see this gastromycetation process, the process of becoming a gasteroid fungus. And so in this diagram, we're showing it becoming a truffle-like fungus. So it's underground or hypogeous. The other element to this evolutionary process is a change in the spores. So in the guild fungi, the spores are forcibly discharged, and over the process of gastromycetation, we lose that forcible discharge. So if we look at this at a broader scale, this gasteroid form has evolved at least 123 times. So many, many, many times we see this process of gastromycetation where the spore-bearing structure is produced enclosed inside something. And so fungi are notoriously famous for convergent evolution, meaning the same form evolves more over and over and over. And that's what we're seeing here with these gasteroid forms. And so just a note on this ballistospore. So if we're thinking about basidiomycetes reproduce via basidia and basidiospores. So we've got these spores that emanate from these little structures called sterigmata. And so what's happening on the base of this sterigmata is we're developing this little water droplet. And at a certain point, that water droplet is going to shoot down that spore and make it forcibly discharged. And so what we're seeing in this shape of this mushroom is we have this asymmetrical shape. The flat part here is hydrophobic and the tip is hydrophilic. So at a certain point, this huge droplet is going to run down this edge of the spore and make it shoot off. So we have forcible discharge. Now, if you're a gasteroid fungus, it doesn't make sense to have that because if you shoot your spore off, it's just going to stay contained in that gasteroid structure. So gasteroid fungi don't have ballistospore, so they need to rely on other mechanisms to discharge their spores. So they rely on different mechanical. Sorry, that is a little glitched up on my end. In this video, here we go. Oh, it doesn't want to play. There's what we're seeing is a drop of water hitting that structure, and that's forcing the spores out. So instead of ballistospore, they're going to rely on some mechanical disruption, a water droplet, or something hitting it, or a twig falling on it, or an animal walking by, or of course us walking through the forest and bashing on them because we love to see them spew out their spores. So if we look at the shape of gasteroid spores versus lamellate species, we're going to see a change in the shape. So the ballistospore, again, has this asymmetrical shape, whereas most gasteroid fungi are going to be round because they're not relying on those dynamics of a ballistospore. All right, so now let's talk about Tulostoma and the morphology of Tulostoma. So here we have a smattering of different species. So let's go into some of the terminology that we use to describe these species. So on the left of this image, we have a primordium, and so that's the earliest stage of development. So at this stage, the whole structure is completely enclosed in this layer of tissue called the exoperidium. So exo means outside and peridium means pouch, and it's the layer of tissue that's surrounding the reproductive tissue. If we go from the exoperidium, the next layer of tissue is the endoperidium. And so over the development of this mushroom, that exoperidium is going to slough away and leaving the endoperidium. And the endoperidium is containing the gleba. And so the gleba includes the spore-bearing tissue. As these mushrooms develop, there's a stipe or a stem that elongates and lifts this whole structure up. And then at a certain point, there's an opening, that stoma or mouth that forms. So if we apply those words to the actual mushroom that we're looking at, or puffball, that top, again, is the stoma or mouth. And when we see one of these, we're looking at the endoperidium because the exoperidium is mostly removed. And you're going to see the exoperidium kind of at the base of this structure. Inside the endoperidium, we're going to see the gleba. And of course, it's elevated by a stipe. And of course, we're mycologists, we're just going to have more words to describe things. So if we take the whole structure, the stoma, the endo and exoperidium, and the gleba, that whole thing makes the gastrothecium. So gastro means stomach, and thecium means hymenium, and hymenium is just a fancy word for that spore-bearing structure. So we have all these words to describe the morphology or shape of these mushrooms. To distinguish Tulostoma, one of the first things you're going to look at is the morphology of the mouth or the stoma. And of course, we've got to have more words. So we call it the peristome. So peri means around. So it means the whole structure around the mouth. So there's basically 3 different structures that we're going to see in Tulostoma. Some of them have this definite tubular shape. And so it's projecting from the top of that gastrothecium. Some of them are eroded and they aren't definite edged, and that's an irregular one. So it kind of just sloughs open. It could be split. It could be somewhat circular. And then the third shape is fibrillose fimbriate. So fibrillose means covered in thin fibers, and fimbriate means fringed. So these are kind of the 3 shapes that we can use to classify our Tulostoma into different groups. And then we have the exoperidium. So that exoperidium, again, is that layer of tissue on the outside that generally sloughs away as it matures. So if we zoom in on the middle here, we can still see some of this tissue layer remaining right there. And this kind of peels off in a sheet. So we're going to call this a membranous exoperidium. And there's different thicknesses to this exoperidium. Some species are going to have a thin membranous exoperidium. Some of them are going to have this thicker membranous exoperidium. And we can contrast this exoperidium or a membranous exoperidium with a hyphal exoperidium. And a hyphal one, it's a little bit confusing because all fungi are made up of hyphae, but in this one, it doesn't form those sheets that peel off. So here you can kind of see some remnants of the hyphae that are running around. So it'll remain maybe a little bit longer than the membranous one that peels off, but this one will also slough away with development. If we go inside to the gleba, to the spore-bearing mass, we've got spores, and we also have these filaments, these hyphal filaments that are called capillicea, and sometimes these can have taxonomic significance. So if we look at this capillicea, there's going to be a wall on some of them, and so that's called a septa. And you want to look at the thickness of the capillicea. So here we have an example of what's considered a thick cell wall. And then the characteristics of the septa. Is it inflated? And so you'll have to measure capillicea, look for branching, those sorts of things. So if you're looking at all puffballs are going to have some sort of capillicea, and there's a lot of variation that you'll see within capillicea. So I mentioned there's thick-walled capillicea. There's also thin-walled, which is represented in panel B. And then if we look at how they break up or how they branch, that can also give us some taxonomic information. So in C, we have Calvaceae type capillicium. And so what we're seeing are a bunch of fragmented pieces of capillicea. And so what's happened there is at those septa, at those cell walls, these capillicea have broken off. So we call that disarticulating. In D, what we have are these main arteries that kind of branch and taper off. So we have these thick capillicea that become thinner as they branch, and those are considered Bovista type capillicea. In E, we have branching, and so what we have here are very even branches, and they're dichotomous, meaning they branch into 2. So this is considered an intermediate type capillicium. And then over here in panel F, we have what's called lycoperdon type Capelicia. So they branch, but it's irregular. So it's not this evenly spaced. They don't all branch into two. Here you can see sort of just a random branch here. So the different morphological features of the Capelicia can be somewhat taxonomically significant. And then, of course, my favorite, the spores. So we see everything from no ornamentation to just crazy ornamentation. So here we have examples of some smooth spores, so no ornamentation. In the next few panels, we see slightly roughened. And then we get into all these different types of spines. So once again, we have to use 100 different words to describe what we're seeing. So we've got things like verrucae, which means warts. So you can think of them as rounded spines. We've got verrucose, which just means a lot of warts. And that's not enough to describe a lot of warts. We're going to use verruculose for a lot of warts. We've got ecanulate, which means spiny. Sometimes those spines start to fuse. So we get these pyramids type structures forming. And so we're seeing that kind of in O and P. And then one of my favorites are these ridges that form spirals. So you can kind of think of the ornamentation starting to fuse and make these spirals. And then even some of the ornamentation forms this kind of reticulate structure around.
Can I interrupt you for just a moment? Going back to the Capelicium, Ken is saying he was trying to ID a Tulostoma.We'll, and it talked about there being crystals on the Capelicium. Do you have a photo of what that looks like?
I don't. So Brumali is generally a European species. I just looked, and there's 2 or 3 collections of Brumali in iNaturalist from Eastern North America that match Brumali sequences. And I'm going to talk about the difference between Brumali and simulans. I have never studied what I believe to be Brumali, so I have not seen incrustations. I've seen some pictures of folks talking about it. And some of those images look a little bit suspect to me, like it might just be some chunks from whatever reagent they're using. I think that lactophenol blue is a really good staining reagent to use when you're looking at some of these Capelicia structures. And that's what this one is stained with. And please let me know if you ask a question and I don't fully answer it for you, and I will try and clarify.
He says, thank you. One of those sequences is his.
Oh, cool. Is it from New York? We could carry on this conversation maybe offline.
Canada.
Canada, cool. All right, Prince Edward Island?
Yes.
Okay, cool. I did just see those, so I'm interested to we'll touch on that. So I'm super interested to throw that in a tree and see if it really is Brumali or maybe simulans or maybe even something different. So we can't talk about Tulostoma without talking about Jorge Wright or George Wright. So he was an Argentinian mycologist and he published a world monograph on it. So he traveled all over the world studying Tulostoma collections. And in his monograph, there's over 200 species that he describes in his monograph. A super interesting guy. So he graduated from the University of Buenos Aires in 1949 after he studied the taxonomy and systematics of Argentinian gastromycetes. He then went to University of Michigan for his master's degree and studied under Alexander Smith. And then he returned back to Argentina and completed his doctoral degree. And he remained at the University of Buenos Aires for 30 years as a faculty member. So he established their fungal culture collection, and there's over 2,500 fungal cultures in his collection, and his fungarium has over 18,000 collections. So here's a picture of him in 1975 from one of his forays in the Parque Nacional Iguazu in Argentina. There's a lot of other folks that have worked on Tulostoma, a lot of South American folks, and I didn't go into all the history of that just for time, but there's a lot of folks that are working on these fungi, and they're super cool. So maybe the most comprehensive phylogeny that we have to date is from Jepson in 2017. And so in their phylogeny, they found 30 named species and 27 unnamed species. So these are some of the things that I was working with when I was trying to identify the fungi that we find in southwestern US. And recently, a great text has come out from Noah Siegel and Christian Schwartz, Mushrooms of Cascadia. So of course, with every new ID book, I always flip to Tulostoma to see what folks say about Tulostoma. And I thought this was great. Of course, they put no name on the Tulostoma species, but in their comments, they describe what it's like to identify Tulostoma. So recognize them as a genus is easy, but putting confident species level names on them is downright impossible. And, you know, it's sort of true, but with the help of scanning electron microscopy and DNA, we're getting a little bit closer. So in my phylogeny, I found about 70 species. I was able to confidently name about 45 of them. There's still about 25 that are unnamed, and we even have some soil isolates. So my phylogeny is based on ITS, LSU, and transcription elongation 1 alpha factor. And so I thought that because they are so difficult, maybe I would take a phylogenetic approach to trying to understand the diversity. And if I could group them phylogenetically, maybe there'd be some morphological characters that were synapomorphies that held them together so I could understand them a little better. So I'll give you.
Is a light microscope sufficient to recognize the same features of the spores? Or do you have to use a scanning electron microscope?
Sometimes. So we'll go into some of, the common species and some lookalikes, and we'll look at some of these SEM pictures side by side, so you can kind of see or imagine what these would look like under a light microscope in the morphological characteristics, and kind of guess for yourself or gauge for yourself if you'd be able to discern that morphological feature under a light microscope without that three-dimensional aspect that the SEM gives us. So
Thank
the
you.
short sometimes, rarely, maybe, those kinds of things. So yeah, I was like, if I make a phylogenetic tree, everything's going to make sense and it'll be easy to group things together, but it's fungi and there's convergent evolution, and they said they're not going to follow any patterns. So this is a good example of how sometimes phylogeny does not help us at all. And I will start this off by pointing out that there's not a lot of bootstrap support for this clade to begin with, but it's just a pretty good example of how phylogenetics doesn't always help us in our pursuit of understanding fungal biodiversity. So in this clade, we'll call it clade 6, if you look at the spore morphology of this group of purported to be closely related species, the spores go from completely smooth to highly ornamented to somewhat reticulate. And then if we take that a bit further and look at the other morphological characteristics, we have groups with membranous and hyphal exoperidium. We have every mouth morphology on these, and again, every different spore morphology. And even geographic distribution doesn't really help us to discern these. So phylogenetics did not really help understand this. And so when I was putting this talk together, I was trying to figure out a way to describe this diversity. And so what I feel like maybe the easiest way to come at it is to start with the most common species and then talk about lookalikes that you might be able to, maybe with a little bit of morphology and some DNA and maybe a scanning electron microscope, differentiate these species. So Tulostoma fimbriatum is by far the most common species, the most commonly misidentified species in this group. And then we also talked about bromeliads and simulons already for a second. And so that'll be an interesting group to talk about as well. So whoops, sorry, little spazzy finger there. Oh, sorry. All right, so we've got Tulostoma fimbriatum. So this has that fibrillose fimbriate cap. And so this, most of the collections that I have, I studied from herbarium collections. So there are a few pictures that I've taken off iNaturalist, and there are some folks that I was not able to contact. So I hope that I've cited your images on a couple of these. So please let me know if you find that problematic. I apologize. But here is a great image of Tulostoma fimbriatum with the fibrillose cap and these really cool spores. So what's happening here is we've got these little spines and they're starting to form these ridges. And so this mushroom has a global distribution. So we find it everywhere in all kinds of habitats. So people are very keen to put this species name on this group. The morphology of the spores can help, but you can imagine how a lot of the mushroom spores that we see in Tulostoma might look like this spore morphology underneath a light microscope. So if we look at the phylogeny of this, just the number of collections that we have in the phylogeny points to how pretty common this is. But if we start to look at the geographic distribution, we start to see a little bit of pattern happening. So we definitely have a clade from North America. We've got this oddball from Russia, and we've got this group from Europe. Then at the bottom, we also have this other group that is somewhat distinctive from the other groups, and it does have some bootstrap support. So when we start to look at the morphology of these, there's so much overlap that there's not a really good morphological distinction. There is a good geographic distinction, but there's not really a good habitat distinction. So most of us who are studying this are putting all these groups or all these clades together as fimbriatum. And as you might imagine, a mushroom that has such a big distribution and a little bit of morphological variation has a ton of synonyms. So this is one of those examples of a mushroom that just keeps getting renamed and renamed and renamed. And just to hammer home the point of this distribution, Andrew Cannon sent me this collection that he found in his flower pot growing on his balcony in Manhattan. So this mushroom really does grow everywhere. It has that fimbriate fibrillose cap and ornamented spores and a hyphal exoperidium. So we have this other kind of clade down at the bottom that seems somewhat distinctive, and it does have some bootstrap support. And I studied the type of it, and so macromorphologically, it's very similar. The spores are a little bit different. And so kind of what I'm thinking here is that this is an old collection, and I think what has happened is the morphology of the spores has started to decompose. So we see that it has some sort of ornamentation that looks like it might be forming these ridges, but if we look at it from different angles, it started to just kind of collapse and turn into blobs. So different authors have made different comments on this. Based on genetics, I don't think of this as necessarily a distinct species unless we can come up with some more evidence to make it distinct or to convince ourselves that it's distinct. A very similar species that also has a fimbriate fibrillose cap, very similarly ornamented spores, and an exoperidium that's hyphal is Tulostoma polymorphum. And so the big distinguishing feature here is the base of the stipe. And so we've all hammered home the point that you need the entire structure to be able to identify. And this base of the stipe is often the thing that we lose as we're extracting it out of the soil. So this is a great example of we really need the base. We have this super flared base at the base of polymorphum, and that's going to be our distinguishing feature. There's a couple of other subtle features. So the spores of polymorphum are 4.5 to 6 microns, which is a tiny bit smaller than fimbriatum, which are 4.5 to 6.5. So in my mind, that's not really a good, easy way to distinguish these. The capillicea can be a little bit more distinctive. So the capillicea and fimbriatum are a little bit more swollen at the septa, so that septa wall is a little fatter, and they're a little bit more branching. But
For me,
really,
sorry, go ahead.
go ahead.
Is that base considered a vulva, or is that just a
It is.
abrupt base?
It doesn't have that piece of tissue. So I'll talk about Chlamydopus at the end, which does have a vulva. None of the Tulostoma that we've seen have a vulva at the base. It's just a flaring base. So we haven't seen a tissue there. And so again, you could see how we had that question earlier. If you were to compare these two spores under light microscopy, you would be very challenged to differentiate the level of reticulation on fimbriatum compared to polymorphum. So here's an example where light microscopy probably wouldn't help you, but again, we've got this flared base that should be pretty distinctive. Another similar species is Tulostoma punctatum. So here we have a different spore morphology. Pretty much everything else is the same. Punctatum lives in the same habitats. It's the same size. It's got a fimbrillose fimbriate cap. It's got a hyphal exoperidium, but the ornamentation is a little bit different. So we have these spines or warts that are rounded, whereas here in fimbriatum, they're a little bit more isolated and a little bit more ridge-like. So here's an example that light microscopy might help you in differentiating these two species because the spores are subtly different. Moving on to bromali versus stimulons. So these two mushrooms morphologically are very, very similar. Historically, it was thought that bromali was restricted to Europe, and Wright thinks that simulons is native to North America, and everywhere else that it's found, it was introduced to. And so if we compare the morphology of these, both Wright and myself think that simulons has a membranous exoperidium. Jepson reports it as hyphal, but all the other morphological features are similar. So they have a tubular mouth. Their spores have the same kind of ornamentation. They are found in many, many different habitats. And you'll notice again that the spore size for simulons is negligibly smaller than the spore size of bromali. So if we look at the phylogeny of these, they're definitely distinctive and they have high bootstrap support. So for those, and I didn't run this, I haven't pulled those bromali sequences out from North America, from Canada, and there's one in New York that's on Non-Naturalist. So I'd be very interested to see how they fall out in this phylogeny. And it'd be an interesting to look at. Is it a new introduction into North America, or have they always been here? So I think that's great, and it's great that you guys have collections of it, so we should definitely run a tree and see where it falls out. As I mentioned, I haven't studied bromali, so I don't have any SEM images of it or any macro photos of it, but it is very similar to simulons with this projecting mouth, a membranous exoperidium, and these ornamented spores. Another species that you might confuse simulons with is the Purpussii. So to differentiate Purpussii, what we're seeing is the spore ornamentation is somewhat reticulate, and then the basidiocarp is a little bit more darkly colored. And this is another example of, I think that this is morphology that spore morphology that you would be able to find a distinction with light microscopy. One of my favorite Tulostoma is in this group, Striatum. So Striatum and Pulchellum are two species that look very similar, but we can distinguish based on their spore morphology. So these both have the fimbriate fimbrillose cap or mouth, ornamented spores, and a membranous exoperidium. The Pulchellum have these warded spores, and you can see some of them are starting to form ridges. But we can compare this to the macromorphologically similar striatum, and this one forms spirals. So these two mushroom spores you can definitely distinguish underneath the microscope.
Can you give a very brief touch on what bootstrap values are? We have several people who don't understand the term.
Yeah, so when you're doing a phylogenetic analysis, and I can't go into the math behind it necessarily, but what you're doing is you're taking a bunch of DNA sequences and applying different statistical analyses to them to replicate purported processes of evolution. And so through this process, it generates an evolutionary tree. And it reiterates this process many, 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 many different times. And so the simplest way to think about it is the number of times that this outcome is generated through the analysis. So one way to think about this is that in 93% of the trees, this shape came out. In this, 70% of the time, this kind of configuration of branches came out. And then this is saying 100% of the time this came out. So 100% is a very high bootstrap value. We generally don't report anything that's less than 0.6 or 60%. And please continue to ask me if that didn't clarify. So it's a level of confidence that we have in, is this tree right? Is it true? Is it a correct depiction of evolution? And I do also want to say, when we're looking at phylogenies, it's always going to be a hypothesis because we were not there. We'll never know the absolute truth, but our ability to reconstruct evolutionary histories is improving as we better understand the data that we're using, and we're using better data to make these hypotheses or estimates. Oh. Okay, and so we have Tulostoma opacum, which have these really cool reticulate spores. And so this is another one that the combination of characters, you'd probably be able to discern this one. So it's got this indefinite mouth. So kind of the stoma, the opening is eroded. It's either like split or cracked. It doesn't form a nice circle, or it doesn't have those fimbriate characteristics. And the reticulate nature of the spores will help you identify this guy. Also through our work, we found a couple new species that we have yet to name, and that's because we don't have a ton of collections of it. So Jepson found two species, 10A and 10B, and I found two collections that match each of his collections. So now for 10B, we have two collections, and it's really interesting because this, I think it's Hungary, is this guy. Whoa. Don't know what's happening. So Hungary, this was found in, and I think that this one was from Arizona, and it has this very distinctive radicating stipe, a definite tubular mouth, and these ornamented spores. And we only have 2 collections, so we haven't described this species yet. We're hoping that we'll be able to find some more of this collection. And then this other species that also has a tubular mouth and ornamented spores. And so, oh shoot, I should have written this down. I can't remember where this one was collected right off the top of my head. So again, we've got a ton of diversity in Tulostoma that is yet to be described. And just in case you weren't aware that mycologists are crazy nerds, they just recently described this Tulostoma after the Shai-Hulud sandworms from Dune. So this is one of Jepson's species that's been recently described. So we've got Tulostoma Shai-Huludii. And then there's a couple of Tulostoma that you have no problem identifying ever. This spectacular exasperatum that has just this beautiful, intricate, almost doily-like exoperidium. But of course, it's Tulostoma, so there's going to be another species that looks just like it, Tulostoma paratiense, also have this crazy exoperidium that looks like a doily, and it's pretty ornamented. It can be spiny when it's really young, and then those spines kind of fall off. But the cool thing about this is you can distinguish these based on location. So if you are in Southeast Asia, you're going to find exasperatum, and if you're in Southeast Brazil, it's going to be paratiense. So let's talk about a little bit about other arid adapted species or adaptations to aridity. So Astreus is an Earth star, but it's not related to other Earth stars. It's not related to Geastrum, and so Aster means star. And there's about 11 species of this, and this isn't not necessarily an arid species, but it has some cool adaptation to aridity. So the rays of this star have a layer of tissue that's hygrophonous. So that means it's going to absorb moisture. And over time, those rays are going to reflex, and it doesn't quite show it in the video, but those rays are actually going to prop up that gastrothecium. So that's going to happen in the rain. So that fungus knows that when it's raining, it can disperse the spores. And that's going to increase the success of spore dispersal and germination. And so one of the reasons I included this genus in this talk is because I find it oftentimes in Florida, and what we're seeing here is these sandy habitats. So I find a couple different Astreus species in sandy habitats. Another cool arid adapted species is the Myriostoma. So this is similar to Earth stars. It has this ray pattern that can also reflects in rays, or reflects back and enable spore dispersal when moisture is available. And stoma, we already know that means mouth, and myrio means myriad, and the direct translation of myriad means 10,000. So it doesn't quite have 10,000 mouths, but it has many mouths from which the spores are ejected. Another very common desert species is Podaxis, and I'm putting Pistillaris in quotation marks because there's some genetic distinction between what we consider Pistillaris in North America compared to other geographic regions. So if we look at the phylogenies of Pistillaris, we can see tremendous genetic distinction. So both of these 2 studies looked at Pistillaris in North America and found that there's genetic distinction between these, but neither of these studies decided to discern these as distinct species. So for right now, we're still calling it Pistillaris, although it could be maybe on its way to being differentiated into different species. And so what I kind of think about these guys is we're catching them in the midst of evolution. So these populations have been isolated for some time. So we're seeing a little bit of genetic distinction, but they haven't adapted so much that they are incredibly distinct from one another. So in my mind, what we're seeing is this step in the process of evolution. Another arid adapted species that we have is Montagnia. And so you might look at that and be like, well, that is just a really dried up Caprinus, which it is. It's closely related to Caprinus. And what you see in these guys is they kind of grow like that. They're instantly dried up. And instead of deliquescing like a Caprinus, they don't deliquesce, they just become brittle. And so these guys are considered pseucotioid. They've lost forcible discharge, and the gills of these fragment and they blow away. So sometimes the spores just kind of break off, but also they can disperse by just breaking off little bits of their gill. There is not a lot of work done on this group, and there isn't a lot of species diversity. So it's interesting. It doesn't seem like anyone's really generated phylogeny of these groups. So if you're interested in looking at phylogenetics, this would be what I consider a low-hanging fruit. There's not a lot of species diversity. So there's probably about 7 or 8 species. So somebody should really make a phylogeny of these and look at the evolution of some of these desert fungi. One other group I'm really interested in is Decidia. And so these I've been collecting a lot because I see it and I think it's a Tulostoma, but it doesn't have a stipe. So it has this same kind of exoperidium at the base. You can see similar tubular morphology, irregular morphology, a fimbriate morphology of their mouths. They just don't have a stipe. And so I've been collecting these. I haven't spent any time looking at them yet, but there is a paper from 1975 that looked at the Colorado species, and they're claiming that these two spore morphologies came from the same fruiting body. So within one puffball, you have smooth and ornamented spores. So what I just wanted to point out is these are habitats that we look at that you might not think that there's a ton of diversity. So this past fall, I got to spend a lot of time in the Pacific Northwest, and I was driving back home to Colorado in early November, and it had been wet. And this is called Devil's Canyon. So I just stopped and got out to go for a little hike, not thinking too much, but poking around. And lo and behold, I found Southerella, and I'm calling it Ammophila. Please correct me if you think I'm wrong. But this is the first report of this in Colorado. No one's collected it. And based on the amount of collections, you might think it's rare. In my mind, I don't think that this is a rare fungus. I just think that nobody's collecting in these areas. So I think these dry, arid environments are rife with fungi that we are just not looking at.
Regarding these dry environments, Jorge is wondering if Tulostoma is a derived genus, and in terms of deep time, how old it could be, and if it evolved along with the deserts.
So one of what I looked at was, and I'm blanking on the time frame, but they evolved from, and I didn't put these in this talk, but it seems like maybe they evolved the same time as the Eurasian steppe. So they arrived, or they were derived at that same time that this grassland habitat was evolving, kind of the same time as when grasses evolved. And that diversification time frame overlaps the diversification time frame of some of these grassland habitats.
Thank you.
And I'm sorry, I should have that number in my head, but I don't have it in my head right now. So I just wanted to spend our last few minutes talking a little bit about fungal biodiversity and conservation. And so I talked a bit about Tulostoma diversity, and I mentioned that we had a lot of diversity of Tulostoma in the southwestern US. So we have 61 reported species. Currently, I've only found about 15 of these species. So why can't I find them? And so one of the reasons, right, is we're just not going in these habitats. If you look at these sagebrush, these grasslands, these desert ecosystems, as mycologists, we're not spending a lot of time in there, right? They're not enticing to us. Another big factor is climate change. So these habitats are experiencing climate change perhaps at a faster rate. So they're the extremes, right? The drier dries, the hotter hots, changes in phenology of the organisms that live in these habitats. And I also want to just touch on public perception of climate change for a second. So on the left hand, we're seeing a graph of people who think or accept the science of climate change and understand that climate change are going to affect people in the US. And so the warmer colors, the redder colors are higher percentages, and the bluer colors are lower percentages. So if we look at the map on the left, most people understand the science of climate change and that it is going to probably affect people in the US. However, on the right-hand side, that map is telling us that we don't think it's going to affect us. So we have this really interesting disconnect that we accept the science of climate change and that it's going to affect America, but we're fine, it's not going to affect me. So moving back to Tulostoma and why I haven't been able to find them all, there's also this factor of habitat degradation. So this fence is separating a grazing pasture on the right from a non-grazed pasture on the left. And you could see that there's been some habitat degradation, and that's happening throughout the world. And then, of course, we have anthropogenic development. So here's a Google Earth time lapse. Don't know why my videos are not working today. There we go. Or maybe not. So as you might imagine, if this video was working, we can see the development, the anthropogenic development over time. And this one is Albuquerque. So over time, we are moving into different habitats. So you might imagine if we have a specialist species, a species that only lives in a narrow little range, if we move into that range and we develop on their land, we're not going to see that fungus because it only lives in the one place and we've taken away its habitat. Oh, now it's working. So here is our little time lapse. We can see development as it moves out into just basically urban sprawl is what we're seeing. And so we really need to be able to document biodiversity to be able to conserve species, because we can't conserve what we don't know. And so we need to think about fungal biodiversity and what we know about it. And so each of these images is the relative size of the biodiversity of each of these groups. So of the estimated about 80,000 animals that exist, we have described about 74,000. So we've described about 92% of the animals that exist. Likewise, for plants, there's an estimated 450,000 species of plants, of which we've described about 400,000. So we know about 88% of plants. Of course, there's a ton of biodiversity of invertebrates. There's an estimated 8.5 million invertebrates, and only about 1.5 million have been described. So we know about 18% of invertebrates. For fungi, the estimates widely vary. Anywhere between 2.5 to 6 million species of fungi exist, and we've only described about 155,000. So that means we only know about 2.5 to 6% of fungi. So we really have a lot of work to do to understand fungal biodiversity, and we are doing a lot. So NAMA does great work. At their annual forays, we go out, you guys go out, you collect fungi, you bring it to the vouchering room, you fill out those annoying little slips, and then we go through and we collect all that data. So we're making species lists, we're counting abundance, we're accessioning them into the Chicago Field Museum. We're doing DNA sequencing. Dr. Andy Wilson gave a talk a few weeks ago on that whole process. So look that talk up if you're interested in it. And so just these processes are so essential for us understanding biodiversity. And so to conserve biodiversity, one of the international groups is the IUCN, and they have developed the Red List. So the IUCN was founded in 1948, and the Red List was founded in 1964. And the Red List is an inventory of the global conservation status and extinction risk of species. Fungi were just added to this list in 2013. So it took 49 years for fungi to be included into the IUCN Red List. And so since 2013, 818 fungal species have been assessed for conservation status. So I just mentioned how much we know about fungal biodiversity. Of the species that we have described, we've only assessed 0.5% of that. So very, very few. So of those 818 species, 340 of them have made the list. So that means 42% of the fungi assessed so far are either critically endangered, endangered, or vulnerable. So we have a lot of groups that are coming at this idea of conservation in different ways. So the promotion of fungi into conservation plans has been spearheaded by a group of people, and Juliana Furci has been very successful in getting the word out and has established fungal conservation in Chile. Most recently, a group attended the Convention on Biological Diversity, and so Chileans and mycologists from the UK called on the UN to include fungi in the conservation for fungi. Closer to home, we've got the Fungal Diversity Survey, and so a lot of folks that are volunteering for that are working on assessing fungi for the Red List. And in fact, there's a group of folks meeting tonight to try and assess fungi for their extinction risk. I am working with a group in the Colorado Natural Heritage Program to try and streamline our databases so that we can get stuff listed on NatureServe, which is what natural heritage programs use to monitor species abundance and diversity for the goal of conservation. You are the first to see our list for the Fundus Rocky Mountain Rare Fungi Challenge. So here's our list. We've got some folks that are working on the media for this, but here's our list, and I shamelessly helped put this together. So I've highlighted 2 of the species that I'm very much personally interested in finding, Schizostoma and Chlamydophis. So Chlamydophis, these are both stalked puffballs. So sorry about the bias, everybody there. But Chlamydophis is a really cool stalked puffball that is not closely related to Tulostoma at all. It's got this crazy expropridium that's just like super gnarled. And here we go with the vulva here. And then Schizostoma. So I have never seen this before and I want to see it. So anybody who finds schizostoma, the first person who sends me a schizostoma, I'm going to buy your NAMA membership for the next year. So get that to me and you get a free NAMA membership. I wish I was Oprah and could be like, you get one and you get one, but alas, I'm a public employee, so just one.
Is that only for Rocky Mountain area for your target
Nope.
range, or can that be one from outside the
Anywhere.
range?
Anywhere. I've never held one in my hand, so anyone who wants to send me one. Oh, but the thing is it can't be in your current collection. I mean, you can send me one, but I want people to go out and keep collecting. So I really want to encourage you to go into these habitats. They're super cool. There's a ton of cool diversity. And if you look at, so I'm in Colorado, so I'm going to use this as an example. You can see where we're collecting fungi, where there's huge population areas, but in these arid environments that are a little less populated, and especially in eastern Colorado, which is the start of the Great Plains, it's dire out there. We don't have enough collections from those areas. So I don't want you to listen to Edward Abbey, who says, stay out of there, don't go, stay home and read a good book. Desert solitaire, for example. The Great American Desert is an awful place. People get hurt, get sick, get lost out there. Even if you survive, which is not certain, you will have a miserable time. The desert is for movies and God-intoxicated mystics, not for family recreation. What I want you to do is think more about how Tom Robbins thought about the desert and desert adaptations. So we have weird stuff in the desert, and he noticed that there's weird adaptations to deal with the desert, such as the camel. And so the camel has a big, dumb, ugly hump. But in the desert, where prettier, more streamlined beasts die quickly of thirst, the camel survives quite nicely. As legend has it, the camel carries its own water stored in its stupid hump. If individuals, like camels, perfect their inner resources, if we have the power within us, then we can cross any wasteland in relative comfort and survive in arid surroundings without relying on the external. Often, moreover, it is our hump, that aspect of our of our being that society finds eccentric, ridiculous, or disagreeable, which is all of us mycologists, I think, that holds our sweet waters, our secret well of happiness, the key to equanimity in malveant climes. So think about Tom Robbins, RIP, as you go out into the deserts and collect fungi and send me your schizostoma. So
Thank you so much, Amy. I have a couple of questions for you. We have a couple of people wanting to verify these are all saprotrophic, correct? So what are they eating? What are they breaking down if they're growing on the sand?
All right, so that is an excellent question. So one of the other things I did look at was their nutritional profile. So if you look at the ratio of carbon and nitrogen isotopes, it can give you some inkling what they're doing. And so they are presumably breaking down whatever limited organic material is in there. However, there is a suite of Tulostoma that appear to have some biotrophic signatures. Now, it's a chapter that I haven't even tried to publish because in my mind, if they were associated with something that was living, we would have picked them up somewhere in some of our root tip analysis, and they've never been picked up. So I did try and get at that question, and I do have confidence that a lot of the species are saprotrophic based on the isotopic analysis, but perhaps not all of them. And they just are pretty good at tapping into some or whatever organic material is in there, just like all of those other arid adapted species like the Montagnia and Podaxis. They're all saprotrophic. They're tapping in to probably things like grass roots and things like that, things that have a high turnover in their organic material.
Is there a possibility that they are collecting mineral content similar to how rock lichen do?
It's a possibility. The fungi that we find that are excreting those kind of crazy chemicals have different hyphal characteristics. I would be surprised if Tulostoma was doing that, but I would not rule it out.
Excellent. In discussion about coastal and sand dune species, there is some thought that some of these dune species have elongated spores compared to their counterparts. Do you see this at all in desert species?
I haven't gone down the rabbit hole of comparing spore morphologies of desert adapted species to their sister taxa. Mostly what we're seeing are pigmented spores with high ornamentation. So those are the ways that they're dealing with issues of desiccation. So having melanin to protect them from sun and ornamentation, presumably to protect them from desiccation.
Speaking of desiccation, how long are the spores viable in a dried fruiting body? And how does Rodney wants to know if being a puffball on a stipe gives it special advantages in the desert.
So one of the things I was investigating was the evolution of spore morphology in these groups. And so I was really interested in the natural, the selective pressures that were leading to these crazy changes in spore morphology. And so I was looking at how maybe different ecological conditions would drive changes in morphology. So I looked at how the ancestral state reconstruction, so meaning what was the ancestral state, what did the spore look like when this genus evolved, and how did the evolution of spores occur over time. And so what it looks like is the ancestral state, so when this genus evolved, it looked like those spores were fairly ornamented. And over evolutionary time, we've lost spore ornamentation. So as trying to get to the answer to your question, Rodney. So the idea there is all these different types of spore morphology, can we look at those and think about or come up with some signal from, so either ecological, selective advantages, or maybe distribution. So if you're smooth spored, are you traveling further? If you're ornamented, are you traveling further? And so none of my analyses were able to discern any different patterns. So, if you're elevated, are you able to travel further? Those sorts of things. I wasn't really able to find any signals. So what I think is that there's more about what they're eating, how they're getting their nutrients, has more to do with the ecology than some of these other structures. Does that answer the question?
It sounded good to me. Speaking of spore ornamentation, Tom wants to know what stains you recommend for studying the microscopy of the spores.
The spores are pigmented, so you don't really need stains. You can see them quite well. The Capelicia, the lactophenol-blue is a really good one. And you can see morphology with that lactophenol-blue. They're not, oh my gosh, this is my third presentation today. When you put the milk, they're not amyloid. So that doesn't really help. And going back to another question about the viability of the spores. So I never try to germinate them, but because they're pigmented and ornamented, in those SEM pictures, you'll notice that some of them collapse a little bit, but generally speaking, they hold their shape for a very, very long time. So they're fairly easy to work with, and there's a ton of them. So just like with all the puffballs, you just tap them and you can get some spores onto your slide and look at them pretty easily.
Excellent. Sego says they are doing a loop around near the Petrified Forest National Park, Navajo Park, some other areas, hoping to see these. There aren't very many observations in that area of Tulostoma on iNaturalist, and they are wondering if you have any tips for where they might look to find them.
It's so random when I find them. It's always when I'm not looking for them. And it's always after a period of moisture, but they are quite long-lived. So oftentimes, like you'll find fresher ones, but going back to that longevity, they're kind of already dried when they're out, so they can live for a long time. And it depends on the species. So like Brumali simulans associates with mosses, so you're going to see them with like small bryophyte type plants. If some of these other species, they thrive in disturbed habitats, so you're going to see them. So I see them anywhere from like associated like very tightly underneath sagebrush to just in like a dirt lot that's been completely disturbed.
And when you say associate, you don't mean mycorrhizal associations, or do you? No. Okay. Do you ever find them in burned areas?
I have not ever seen them in a burned area.
Okay. Pat from Virginia wants to know if you can tell us anything about what species we might find on the East Coast, and if they might be associated more with coastal habitat, since we don't really have desert out here.
There is not a lot in Virginia, Pat. Let me pull up the distribution map and kind of
Speaking of that distribution map, we have a request for a link to that. Do you have that you can share?
Yeah, that's Scott Bates built that. If you just Google Scott Bates Tulostoma, it should come up. And if I can pull it up, I will throw it in the chat real quick. There it is.
Is elevation or season a factor with finding Tulostoma?
So if we're looking at the East Coast, you see a lot of blank spaces over there. So you could, like, we've got some in Florida. I found a couple down there, but generally speaking, like, not a lot of diversity there. So if I was looking for it, I would be looking in sort of your sandy habitats. I don't know that there's a lot of like coastal dunes diversity of these species, more grassland kind of things. Elevation. When I think of it, I'm thinking more of habitat than particular elevations.
Being my fungal knowledge is extremely limited to Alabama and the Southeast. And if they are found in Alabama, I would highly suspect that they are found in Georgia and that is a under documentation case. And same with North Carolina and Tennessee, that habitat has awful lot of overlap. I think we need some people to be out looking for these. I think we can give Amy some new material to be studying on these East Coast Tulostoma.
I love it. Bring it on.
Let's see. Brooke, can you just drop that in the chat instead of the Q&A? Bonnie says in Southern California deserts, fungi are more likely in washes where organic debris has gotten deposited deeply over time. Also, do you know if Bacteroides degutii is mycorrhizal with mesquite?
You know, I've never seen Bacteroides, and I haven't studied it, so I don't know much about it. Again, it's just one of these groups that like is very understudied, and it looks like you guys are all jazzed about looking into it. I love it.
We had at one point we had 134 people on this presentation who are now all stoked. You got the fire lit about Tulostoma, Amy. How about scrub habitat in Florida?
Oh yeah. Yeah, so there's a couple of species. I just got a couple of them. I haven't looked at them microscopically. So I have a whole other pile that folks have sent me in the last few weeks, that I'll be getting to hopefully this spring if, you know, the full-time job and all that other stuff doesn't get in the way. But yeah, there's some cool Tulostoma in Florida for sure that I don't think we know anything about really.
Do you have your slides available anywhere for reference, or just we'll have the recording up with NAMA?
I don't have them available.
Thank you.
I have images from other people that I haven't expressly gotten their permission for, so I don't want to share them until I get express permission from some of the images that I've garnered off the internet.
Stolen. I do that too. I'm like, if you see your image, I tried to contact you. Please just let me know if you're mad. Does anyone else have any other questions? We got 7 more minutes. If anyone has anything left they want to say, this was great, Amy. I'm excited to see what we can discover over here in the East Coast for you. I have something like 30 collectors that go out and find things for me in Alabama. I'm going to put Tulostoma on their radars. We're going to get you some Tulostoma.
Yeah, I mean, and it doesn't have to be Tulostoma. Just go out and document fungi, like everything that we're doing. It's all. If you've been to NAMA or any of the amateur club meetings where we're like document, 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 I promise that we're starting to use that data. We're really coming up with some cool pipelines so that we can use that data to understand biodiversity, to try and enact some more protections around fungi. So even if Tulostoma is not your jam, whatever fungi that you're interested in, we're really excited for everybody. One of the things I didn't throw in this is, we talk about biodiversity, and I like to compare mycologists to ornithologists. So there's about 10,000 bird species in North America and about 6,000 ornithologists, whereas there's way more fungi and at maybe about 300 professional mycologists. So if you think about that, each mycologist is responsible for about 8,000 species of fungi. And so we can't do it, and we need everybody. And so it's a pretty cool time to be a mycologist because we have all these folks that know the fungi in their backyard way better than any academic could ever possibly know it. People are jazzed. The community is just a tremendous community. Everybody's working really well together, sharing resources and information. So it's a pretty cool time, and I'm pretty happy to be a mycologist at this point and working with all the people that I get to work with.
Yes. And Amy had mentioned the new upcoming Rocky Mountain Fundus Rare Fungi Challenge, but if you are not in the Rocky Mountain region, there are rare fungi challenges for many other areas in the United States, and I have dropped a link in the chat for the other Fundus Rare Fungi challenges. Go take a look. See, you got a scavenger hunt. Everybody likes a scavenger hunt. Go see what we need more data on, and go out and see if you can find it. Spring is right around the corner, guys. Okay, we have a couple. All right, we have a few more last questions. Have you ever found any truffles in the desert, Amy?
No, I've found a couple around here in Crested Butte, but they were all in coniferous forests.
What is the size range of Tulostoma?
They're about that big.
Are they all roughly that size?
3 to 4 inches. Like some of them will be like if the stipe isn't totally elongated, maybe like less than an inch, but they're not very robust.
Okay. And if people find some Tulostoma, how can they get a hold of you to try and get you their specimens?
My email, I'll put it in the chat. How's that?
Perfect. And Ken says he has lots of specimens of Satharella ammophila. So if you want some to compare against yours, he'd be happy to send you some.
Cool. I'll let you know. I have I have it sitting on my desk right now. And I have a whole bunch of stuff I need to send out for sequencing. So yes, I have mushrooms all over my desk all the time that I will get to sometime real soon.
As is our curse and our blessing and our backlog of fungi. All right, everyone. Thank you so much for joining us. Thank you, Amy. This was wonderful. Everybody, have a good night.
Thanks, everybody. Have a good one.